Have you ever, really stopped to think about the human eye?
Huh. I can't say I have. Not in that way.
Well, think about it. It's wet, it's warm, it's loaded with nutrients, it's constantly exposed to, air, dust, someone sneezing on the train.
Everything.
It is, for all intents and purposes, the perfect petri dish.
A microbe's paradise. And yet, you're right. We aren't all walking around with constant eye infections. It's a bit of a paradox.
Exactly. And I always figured it was just, mechanical, tears washing stuff away, eyelids being windshield wipers. But the paper we're diving into today says, nope, there's more to it. We have an inside man.
Or an inside microbe, to be precise.
An inside microbe. And this is a deep dive that's going to really get into the nitty-gritty of the immune system. We're looking at a brand new study just published this February 2026 in Immunity.
That's right, by Hughes and his colleagues. And what's so exciting here is that we're moving way beyond the simple idea of good bacteria.
Oh, yeah, this isn't just probiotics are good for you. This team figured out the exact, and I mean the exact, molecular password a specific bacterium uses to switch on our eye's entire defense system.
It's a huge step. We're going from biology to chemistry. They didn't just find the bug. They isolated the single specific molecule doing all the work.
Okay, so let's set the stage. The hero of this story is a bacteria called Corynebacterium mastitidis. We can call it C-mast.
C-mast, yeah.
So C-mast lives on our eyes, and we know it protects us from nasty pathogens. But the big question was always, how?
Right. Is it fighting the bad guys hand to hand, or is it, talking to our own cells?
And this paper proves it's a communications game. C-mast is basically training our immune system. But to figure out how, the researchers had to do some pretty serious detective work.
They had to take the bacteria completely apart.
Which brings us to my favorite part, this idea of lipidomics.
Sounds fancy, but it just means studying the lipids, the fats. The researchers had a hunch that the secret password was hidden somewhere in the bacteria's cell wall.
Which is waxy, right?
Very waxy and full of these special lipids. So the first step was to just get them out. They did a lipid extraction.
How does that even work? You just squeeze a bacterium?
Not quite. You use solvents, in this case a mix of chloroform and methanol. It dissolves all the fats and separates them from everything else, the proteins, the DNA.
So you're left with a tube of what is essentially bacterial grease.
That's a great way to put it. A tube of crude but very valuable grease. And when they put this grease on immune cells in a petri dish,
the cells went nuts.
They went nuts. Specifically, dendritic cells and a type of T cell called a gamma delta T cell. They just started pumping out this signal, IL-17, which is a major call to arms for the immune system.
Okay, so they know the weapon is in the grease, but that crude extract has, what, hundreds of different molecules in it?
Exactly. So they had to refine the search. They used a technique called solid phase extraction.
Okay, what's that?
Imagine you have a big jar of mixed nuts, and you want to find only the cashews. You might use a sieve with holes big enough for peanuts, but too small for walnuts. Right?
Yeah, you filter by size.
SPE is kind of like that, but it filters molecules based on their polarity, how much they like fat versus how much they like water.
So they split the grease into different fractions?
5 fractions to be exact. And they tested each one. Fractions 1, 2, and 5, nothing.
They're dead.
The immune cells barely noticed. But fractions 3 and 4, that's where the action was, a massive response.
So the culprit is in there. What did they find?
They found a specific class of lipids called corynomycolates. These are fatty acids that are pretty unique to this family of bacteria, and they narrowed it down to 3 main suspects within that group.
Okay, let's get the lineup. Suspect number 1.
Naked corynomycolates. Basically just the fatty acid tail, nothing else.
Suspect 2.
TCM. That's the fat tail attached to 2 sugar molecules.
And the third.
TMCM. The fat tail attached to just 1 sugar molecule.
And this is the real aha moment of the paper. They tested all 3. The naked fat.
Did nothing. It's like a blank key. No teeth.
Okay. What about the one with 2 sugars, the TDCM? You'd think yours better, right?
So
more visible.
You would, but it also did almost nothing. And the reason is, well, it's about fit. The 2 sugars make the head of the molecule too big and bulky.
Oh, so it physically can't get into the lock.
It can't squeeze into the receptor. The chemistry term is steric hindrance. It's just too clumsy.
Which leaves our Goldilocks molecule. Suspect number 3.
TMCM. Trehalose monocorynomycolate. The 1 lipid tail and the 1 sugar head. It fit perfectly. And this molecule, all by itself, was enough to trigger the entire immune cascade.
That's incredible. Okay, let's pause for a quick recap because this is the foundation for everything that comes next.
Good idea.
First, we have this good bacteria, C-mast, protecting the eye. Second, the active ingredient isn't the whole bug. It's one specific lipid molecule called TMCM.
And third, structure is everything. It has to be that lipid tail with exactly one sugar molecule attached. Not 0, not 2, just 1.
The perfect key. So now we have the key. We have to find the lock.
Exactly.
You mentioned 2 immune cells were involved. The dendritic cells and those gamma delta T cells.
Oh.
Who's got the lock?
It's more of a relay race. The first cell to see the key is the dendritic cell, or DC, the sentinels, the watchguards.
Okay.
And the study found that for a DC to C TMCM, it needs a 2-part lock.
Like 2-factor authentication for your email.
Precisely. The first part is a receptor called MINCL. MINCL is built to recognize lipids that have sugars attached. The second part is another receptor, TLR2, which is more of a general-purpose bacterial sensor.
So the DC needs to see both signals to be sure it's a real threat or a real signal.
Yes, and they proved it. If they used mice that were missing the gene for TLR2, or if they blocked the MINCL receptor, the dendritic cells were completely blind to TMCM.
So the sentinel sees the key, uses its 2-part lock, and confirms the signal.
Then what? Then it shouts. It doesn't fight directly. It's a coordinator. It releases a massive amount of a signaling molecule, a cytokine, called interleukin-1, IL-1.
IL-1. So the DC shouts IL-1, and that wakes up the T cells to go and fight.
Well, that's what everyone thought. The old textbook model was that these particular T cells, this V gamma 4 subset, just needed to hear that cytokine shout. But that's not what the study found.
Oh, wow. What did they find?
They did this really clever experiment called a transwell assay.
Okay.
They put the dendritic cells in the bottom of a dish and the T cells in a little basket hanging just above them, separated by a filter. The filter lets the IL-1 shout pass through, but the cells can't actually touch.
And what happened?
Nothing. The T cells heard the shout, but they completely ignored it.
So they have to physically touch each other.
They have to touch. There needs to be a physical handshake.
Yeah.
And this means the T cell receptor, the TCR, has to be involved.
Which leads us to the most, I think, elegant part of this whole paper, the switch mice. Can you explain what those are?
This is just beautiful genetics. They needed to prove the handshake was vital. So they engineered mice where they could, on command, flip a switch that basically cut the phone line inside the T cell. The handshake mechanism was disabled.
So the T cell is alive, it's floating around, but it can't process the signal from the handshake.
Exactly. And when they flipped that switch, the T cell stopped responding. But here's the crazy part, the real discovery. The handshake isn't the signal to attack.
Wait, what? If the handshake isn't pulling the trigger, what is it doing?
It's telling the soldier to take their earmuffs off.
Their ears.
Their IL-1 receptors, the ears that hear the IL-1 shout. The study found that these T cells need that constant, gentle handshake from the dendritic cell. If they don't feel that handshake, they assume everything is fine, and they pull their IL-1 receptors inside the cell. They literally take their ears off.
That is so cool. So let me see if I have this right. The T cell is a soldier. The DC is the commander. The IL-1 is the commander shouting, "Attack!" But the soldier won't listen unless the commander first walks over, shakes their hand, that's the TCR signal, and that handshake is what tells the soldier to take off their noise-canceling headphones.
That's a perfect analogy. It's an incredibly smart failsafe. You don't want these T cells going off half-cocked just because they hear some stray IL-1. They have to be at the scene, confirming the target with a handshake before they're allowed to listen to the order.
And when both things happen, the handshake and the shout, then what?
Then the T cells unleash the fury. They pump out IL-17, and IL-17 is a signal that calls in the neutrophils.
The heavy infantry.
The heavy infantry. They swarm to the surface of the eye, and they kill any pathogen they find.
Okay, that was a lot. Let's do another quick checkpoint.
Good call.
Step 1. The TMCM molecule hits the 2-part lock on the dendritic cell. Step 2. The DC shouts IL-1 and physically shakes hands with the T cell.
Right.
Step 3. The handshake tells the T cell to keep its ears on to keep its IL-1 receptors active.
And step 4. The T cell hears the IL-1, fires IL-17, and calls in the neutrophils to protect the eye.
The complete circuit. But, this is all in a dish or in a mouse model. Does this actually work in a real-world infection scenario?
The proof of concept. This is where they really sealed the deal. First, they did it by breaking the bacteria.
They made a mutant?
A mutant C-mast. They used a genetic tool to knock out a single gene called FAD-D32.
And what does FAD-D32 do?
It's a critical part of the assembly line that builds the lipid tail of TMCM. No FAD-D32, no TMCM.
So you have a living bacteria, but it has no key.
No key at all. When they put this mutant onto the eyes of mice, It was basically invisible to the immune system. It couldn't trigger any IL-17.
And the mice were vulnerable to infection.
Completely vulnerable.
Yeah.
But then they did the reverse, and this is the part that probably has pharma companies making phone calls.
The synthetic TMCM.
Exactly. They made pure TMCM in a lab, no bacteria, no DNA, just the molecule itself in a liquid, and they put drops of it into the eyes of mice.
It worked.
It worked perfectly. It did everything the live bacteria did. It primed the T cells, it called in the neutrophils, and it protected the mice from a really nasty infection with Pseudomonas aeruginosa.
Wow. Hang on, a safety question.
Yeah.
If you're calling in all these aggressive neutrophils, aren't you just giving yourself inflammation, like a chemical pink eye?
That is the multi-million-dollar question, right?
Yeah.
And then they looked at that very carefully. They checked for pathology, for corneal thickening, for tissue damage, and the answer was no.
How is that possible?
They called it physiological inflammation. Think of it as putting guards on patrol versus having them actually in a firefight. The TMCM brings the neutrophils to the eye and puts them on high alert, a ready state.
So their weapons are holstered until they actually see a bad guy.
That's it. It primes the barrier without damaging it. And the kicker, they didn't just stop with the eye. They realized, hey, this same family of bacteria lives on our skin, in our nose. So they put the synthetic TMCM on the skin of a mouse's ear and up its nose.
And it worked there too.
It worked there too. Same mechanism. It primed the local immunity. This isn't just an eye thing. It seems to be a universal password for our body's barriers.
Which opens up a whole new world of treatments, right? We're moving beyond probiotics
To what we call postbiotics, or MMPs, microbe-associated molecular patterns. Instead of using the risky live bug, you just use the safe, stable molecule.
A molecule doesn't die on the shelf. You can control the dose.
Precisely. Imagine a contact lens solution with a tiny bit of TMCM in it.
That would be a game changer. You could prime your eye's defenses every single night.
Or a skin cream for someone with eczema who's prone to infections. It's about borrowing the intelligence of our microbiome without having to deal with the live organisms.
You know, this whole thing really makes you think about what it means to be an individual organism, my immune system. But this paper shows my T cells will literally take their ears off and stop listening if they don't get a handshake from a bacteria.
It's a perfect example of the old friends hypothesis. We co-evolved with these things. We're not solo artists, we're a superorganism. The bacteria are playing a crucial part of the symphony.
We are, kind of incomplete without them. We're an engine just waiting for a bacterial key to turn us on.
That's a really profound way of looking at it. We are deeply molecularly dependent on our microbial passengers.
So the next time you rub your eye, just remember, you're not alone in there. You've got billions of tiny allies holding billions of tiny keys, all working to keep the guards at the gate awake.
And now we know exactly what that key looks like.
A lipid tail and one single sugar head. Thanks for taking the plunge with us today.
Anytime.
If you want to check out the actual data, we'll have a link to the paper in the show notes. Until next time, keep your ears on. This is The Deep Dive.